And finally, the last method that we are going to see is basically how to use the RIGER or the RNAi gene enriching ranking analysis to identify the hits from the RNA library screen. Now, for the RIGER analysis, unlike the C-score and the RSA, which you can actually use the C-score as the input. Now, for the RIGER, basically, if you look at the Word document file that I sent to you, basically you need to create these four columns in the new Excel sheets. So I'm going to create that basically. Now, so that's in the new Excel sheet document. So I'm going to put it here now. So you will have the reference symbol, and then you will also have two additional columns for your data. One is actually the control, another one is for the treatment. Now in our case, if you remember correctly, so if I go back to the original data set here, basically for each cell lines after you have extracted, you actually have the column name as plasmid and also sample. Obviously, plasmid is actually the control, whereas the sample is actually the enrichment or the depletion of the shRNA following a prolonged period of culture, and this is actually somewhat similar to the treated data. So but for our analysis, what we need to do is basically, of course, these are just the shRNA count. We haven't actually normalized it. Now, for us to actually use a RIGER analysis, we have to go back to the output file that we received from the shRNA seq, basically. So if you go back to the output file that we have, you will actually get a output file from the shRNA seq uh, that looks something like this. I can copy this and then put it into a new Excel sheet for you to see it clearer. So basically you will get a Excel sheet something similar like this. This is actually the output file from the shRNA-seq. Well, remember in the output file or we actually didn't change the header. So the time2 read counts or rep2 basically or basically whatever that's referred to time2 was actually referred to the sample, okay, in our data set, whereas the time1 is actually the plasmid or the control. So then you follow by this column here, you will see time2 normalize, okay. So and then you also have the time1 normalized here. So we will actually use the data, the normalized data from these two columns to input into the input file for the RIGER. So, okay, before we do that, okay, so we are going to copy, okay, for example, this is from BXPC3, basically. So we are going to create the input file here, reference. I'm just going to use the TRC number, all right. And then for the gene, what is the next column? Okay, the next one is actually symbol. So I'm going to copy the gene symbol here and then to put it here. And then after that, I'm going to create this. This will be plasmid, which is the control and also sample. Okay. Now, remember, okay, so don't mix up here. This is the critical part because the time2, this is actually your sample data. Okay, so it will input it into here, and then the time1 is actually your plasmid data. So if you swap it, so then your data will come up directly opposite, basically. Okay, here you go. So you have the first input file for the RIGER. So I'm going to save it now under the certain folder. So I'm going to save it as under the research folder here, results RIGER. So I'm going to save it as input. Now, be careful is that, well, okay, because this was the RIGER was actually created some time ago, so it won't recognize the new Excel workbook format. Instead, what you need to do is that you have to save it under the Excel 97 or 2003 workbook. So you can save it, okay. Okay, once you have done saving it, now we are going to open the RIGER or the what we call it the Gene E software. So you can download it. So in fact, I'm going to upload the Gene E program into the Dropbox. You can actually install it and then follow the instruction given under the Word document. This one, the lentiplex analysis. You have a section called RIGER. It will tell you how you can actually what do you call it, install the RIGER Java file into the GeneE extension. So anyway, so well, what we are going to do now is we are going to run the GeneE. So you can run it here. Okay. So this is the interface of the GeneE. So wait for it. Okay, done. So what I'm going to do is I'm going to the file and I'm going to open file. So this will be the RIGER input file that I just created just now. I'm going to input it. Okay, so here it will basically preview to you the data set that you have, but you need to select which one is the row metadata, column metadata, and the data matrix. So in this case here, the data matrix is basically from these two column onwards. So I'm going to put the mark here. What it says here, basically this is considered row metadata, and the pink color column metadata, and the blue color is the actual data matrix that I have. So I'm going to press OK here. Then it will generate a type of heat map like this. You don't have to bother too much about it. So the next thing what I'm going to do is I'm going into Tools, and then I'm going to run the RIGER here. Okay, so then you will get this interface. So what is important here is basically you need to tell the RIGER or tell the GeneE program which one do you want to compare to. So a notation I will be based on reference. So when you click it, there's two selection here. Basically the class A will be the control, which is the plasmid, and then the class B would be the sample here. Okay. Now there are many metrics for the ranking of the hairpin. In this case here, I normally prefer to use the log fold change. Okay. Number of permutation we will leave it at 1000. Method to convert hairpins to genes. So I'm going to use the second best rank algorithm here. Okay. And then this one I can leave it as it is. So this one okay. All these are set. Basically, I just need to press OK. Okay, it is done now, basically. So now what it does is very similar to the RSA. It collapses all the individual shRNA score into a single p-value that represent in each gene. To look at the data, basically you can go here and then you right click, then you can actually look at gene report here. So that will show you basically the data. For example, okay, in this case here, ITSN2, there are quite a number of hairpins, okay, so you don't have to worry too much about it. But again, the one that we will pay attention to will be the p-value here. If you want, you can actually save this data. Basically, you can save it as output here under the same folder. Then you can open it in the Excel to look at it much more clearer. So I'm going into the RIGER here, okay, and then output. You can open it using the notepad. So you can then copy this into the Excel file if you want to look at that. So that's here you go. So you can see that, okay, this is a gene name. This is the different hairpins that target the particular genes. There are altogether 15 hairpins, and these are the rank of the individual hairpins. And then, okay, the p-value here that you have basically. And this is the most important column that we are going to focus on. And again, once we have done that, we are going to put that into the master data file that we have. We can create one additional sheet here and label as RIGER. Okay, and of course then the gene list, I can copy it here, and then the cell lines I can copy it over here. So the next thing is that you can actually copy the genes and also the p-value here. You can uh, put it into a separate column, and again using the VLOOKUP. Okay, So lookup value will be AVP2. This will be this two column, return 2 and false. Okay. So when you do that, then you copy all the way down. So then you will have your p-value for each of the cell lines here. Okay, so it's almost, well, it's quite straightforward. So by now you should have completed, okay, three analyses. One is using the z-score, whether it's a cutoff or the mean, or you can use the RSA, and finally you should have also the data for the rigor as well. Thank you.